This procedure begins with removing the cerebellum from P4 to P5 newborn mice. Meninges and the choroid plexus are removed from individual cerebella, which are then collected in a 15 milliliter conical tube. The cerebella are dissociated with papain and brought to a single cell suspension with gentle trituration. This suspension is filtered by gravity through a nylon mesh with a 70 micron pore size. The flow-through is gently layered on top of a discontinuous Percol gradient and centrifuged to separate the granule cells from the glia, interneurons, and Purkinje neurons. Cells in the interface of the gradient are collected and pre-plated on poly-D-lysine-coated plates to further remove the glial cells. Granule cells are collected, counted, and plated at desired densities on poly-D-lysine-coated glass cover slips or plastic culture plates.
Hi, I'm Hae-Young Lee from the laboratory of Dr. Lloyd Greene in the Department of Pathology at Columbia University. Today we'll show you a procedure for isolating and culturing cerebellar granule neurons from postnatal mice. We use this procedure in our laboratory to study granule neuron progenitor, proliferation, and differentiation. So let's get started.
This procedure begins with using 70% ethanol in the dissection area. To dissociate cerebellar granule cells, we use the papain dissociation system. When starting a new kit, prepare the albumin ovomucoid inhibitor solution. To do this, take 32 milliliters of Earl's balanced salt solution.